Hi students, welcome all of you to the topic the structure and classification of animal viruses. In this syllabus, in your syllabus, you have to study about different type of viral diseases. Before studying in detail about viral diseases, we need to study the structure of animal viruses. The structure of viruses you might have studied earlier, that we will revise in this session. And then we need to know about the classification of animal viruses, that also you studied in your second semester classes, how the animal viruses are classified. But in your syllabus, you have to study the structure and classification of animal virus. So we are briefly revising this topic. So first of all, we define viruses. Viruses are defined as a large group of submicroscopic infectious agents that are usually regarded as non-living, extremely complex molecules that typically contain a protein coat surrounding a DNA or RNA core of genetic material that are capable of growth and multiplication only in living cell, that cause various important diseases in humans, animals and plants, and also in bacteria. Here in this definition, you can see that that they are submicroscopic infectious agents. That means you cannot see viruses through a normal microscope. We need the help of electron microscope to see viruses. That is why they are called as submicroscopic agents. And they are also regarded as non-living because they need the help of another host cell for the growth and multiplication. They are dependent on the host cells. And another important thing is that they contain a protein coat. Their structure is extremely complex and having a protein coat which includes an core of genetic material which is made up of either DNA or RNA. You know that viruses carry either DNA or RNA, and they are capable of growth and multiplication only in living cells. And they can grow in different living cells like human beings, animals, plants, and also they can infect bacteria. That group of viruses that infect bacteria are called as bacteriophages. And then we will briefly look upon the structure of viruses. Viruses, you know that it consists of 3 parts or sometimes maybe 2 parts. The first part is the inner core, this submicroscopic particle having an inner core which is the genetic material which is made up of either DNA or RNA. And then this genetic core is surrounded by a protein coat, and the protein coat of this viruses is known as capsid. And capsid, it is a polymeric compound which is made up of monomeric unit known as capsomeres. And moreover, some viruses, not all viruses, carry an envelope. They carry an envelope which is made up of lipid and proteins. That is lipoprotein containing lipid envelope will be there. And on this envelope you can see sometimes spikes. And then the spikes and the envelope may carry spikes on this. That is, only enveloped viruses carry this protein lipid envelope. So inner genetic core and surrounded by a protein coat which is blue in color, and which you can see outer layer, that is the envelope, and which may carry spikes. Now we will see the classification of viruses based upon the host cell. So what are the type of host cell they are infecting? Viruses can be classified into plant viruses, animal viruses, or bacterial viruses or bacteriophages. So in this, here we are going to deal more about animal viruses. And animal viruses are classified based upon various criteria. And this various criteria include: one is based on the morphology. Based on the morphology or structure of virus cell, we can classify viruses into different types. And based on the presence or absence of envelope, we can classify viruses. And based on their host preference, based on the in which host cell they cause infection or they attack, we can classify viruses into different types in the host cell itself. And the viruses have got particular cell preferences also. Some viruses will attack only certain type of cell types. And based on their genomic composition, whether they carry DNA or RNA, we can classify animal viruses. And these criterions, that's morphology, presence of envelope, host preferences, genomic composition. This classification is actually it's an earlier method of classification, but the correct classification or the modern classification of viruses is actually based on how viruses make use of its genetic material to make viral proteins, and also its components, viral proteins and then viral assembly, and whether it possesses a double-stranded genetic material. You know that viruses are independent or dependent. They are not independent, they are dependent on host cell. Once they enter into the host cell, they take up the host cell machinery to make up their own proteins. That is, they have only genetic material. By using this genetic material and by using all the enzymes of the host cell, they will synthesize their own proteins and they will produce viral components. And these viral components assemble together to form the mature virions or the infectious virus particles. So that is how they multiply in animal cells. We will see in detail about the multiplication of viruses, but this is actually the way in which they multiply. So during this multiplication, how they use the genetic material to produce viral proteins, and also based on whether they carry DNA or RNA as genetic material. This is an important criterion for modern classification systems. So based on the morphology, we can classify viruses into different types. So based on the morphology, this morphology is based on the symmetry of this nucleocapsid arrangement, and also the morphology of the nucleocapsid and the arrangement of this protein subunit capsomeres. I already told you that viruses are made up of genetic material core surrounded by a protein coat, and this protein coat is made up of simple units or monomeric unit known as capsomeres, and this capsomere are viral components and it has got its own symmetry. You have studied about the protein structure, the different levels of protein structure, and in the quaternary structure of proteins you might have studied about the symmetrical arrangement of protein. So this capsomeres, how they are symmetrically arranged, based on this the virus morphology differ. And you can see that the three important morphological types of viruses, one is known as the rod-shaped virus, which has got a helical symmetry. Here the capsomeres are arranged in a helical pattern. That is why they have got a rod-shaped structure. And the second group is spherical viruses, and spherical viruses usually they have got a particular, this is actually a helical structure, and then this is a spherical structure, and this spherical structure, you can see that these capsomeres are arranged in an icosahedral pattern, icosahedral symmetry. Icosahedron is this is spherical structure which has got 20 faces, just like a football. That's a type of symmetrical arrangement. And some viruses which have got a complex symmetry, which does not have a helical or icosahedral symmetry, but the symmetry will be complex and they will have a complex morphology. So here this is the types of viral symmetry. This is an icosahedral symmetry, which is a non-enveloped icosahedral. You cannot see the envelope covering. Here this is an enveloped icosahedral, and here this is an helical, that is non-enveloped form, and here this is an enveloped helical virus, and this is a virus with a complex symmetry. And the symmetrical arrangement, due to the symmetrical arrangement, viruses has got different morphology, and viruses will be having different size and different morphology. You can see a comparison of the size and morphology of viruses. This is actually a scale of 1 micrometer size scale. You can see different types viruses with different sizes, and you can see the vaccinia virus is the largest virus, which is almost equivalent to the smallest protein, I think smallest bacteria, mycoplasma. And then the smallest viruses, which include the picorna virus, the poliovirus family, picorna virus, and also bacteriophages, are very small viruses. Sorry, this is a type of, this is bacteriophages, and this is actually a particular type of phage. Then this is actually the picorna virus, is the smallest group, and this is the rhabdovirus, which has got a bullet shaped, and this is a paramyxovirus, which has got a spherical shape. So each virus, this adenovirus, has got a different shape, influenza virus, etc. So viruses will be having, especially animal viruses, and also bacteriophages has got a common morphological form, but animal viruses, they differ in the size and shape.